So we continue with our series of lectures on pathology. This is part 3 of a 4-part video on cell injury, adaptations, and accumulations. If you haven't yet subscribed to the channel, what are you waiting for? Grab a piece of paper and let's go. Hello and welcome to MK's Medical Review Series. My name is Dr. Moses Kazevu. This is the series on my YouTube channel where we look at medical topics in depth. Today we're going to be looking at cell injury, adaptations, and accumulations. If you haven't watched the previous 3 pathology videos, or rather 2 pathology videos on cell injury, the first one was on an introduction to pathology, I'll leave the playlist tagged at the end of this particular video that you can actually check out and learn a lot about. In this particular video, we're going to be looking at cell death. Remember, in the prior videos, we looked at cell adaptations and cell injury. And in this particular video, we'll pretty much focus on necrosis and apoptosis. I will release 2 videos today, so I aim on finishing this chapter on cell injury, adaptation, and intracellular accumulations. So remember where we're coming from. All cells are under this constant maintenance of an internal environment, which we call homeostasis. And each of these cells could undergo certain stress, certain injury. If the injury is mild, then it may lead to reversible injury if it's mild and transient. If the injury is severe and progressive, it may lead to irreversible injury, which may result in either necrosis, which I like to call genocide, and apoptosis, which I like to call cell suicide. Alternatively, this injury can cause stress, and these stresses lead to certain adaptations that we talked about in particular cells. And if the cells fail to adapt because the injurious stimulus is still present, this may progress to cell injury. Remember that cell death is one of the most crucial things to happen in the body, and it's going to be affecting many tissues, many organs, and it's going to be resulting from different causes, as we already established in our previous lecture. If you didn't watch that previous video, you may pause this video right now. I'll leave it tagged at the end. I'll also leave it tagged right here so that you can go check out the previous video on cell injury. But it's going to be due to ischemia, which is a lack of blood flow, infections, physical agents, chemical agents, toxins, immune reactions, nutritional imbalances, and many of those factors that I explained in depth in the previous video. Remember that cell death is both physiological, because in the process and development of certain organs, it is actually quite essential for the cell death to happen. For example, in the development of the fingers, it's essential for some of these cells to actually die. And it's going to be a process that's going to also be happening in embryogenesis, which is development of the human being in the different body parts and the different organs and organ systems, as well as development of the different aspects of the body and in maintaining homeostasis. So remember that cells are going to be dying from necrosis, and remember that here the hallmark of this is that there's going to be loss of integrity of membranes. There's going to be loss of the integrity of the cell membrane, there's going to be loss of integrity of the nuclear membrane, there's going to be loss of integrity of the mitochondrial membrane, there's going to be loss of integrity of the lysosomal membrane, such that leakage of some of these contents into the cell cytosol is going to result in activation of certain enzymes. It's going to result in degradation of certain components. And then, of course, this is all in response to this injurious stimulus that was there. And remember that some of these enzymes are going to be found in the lysosomes, and the cells are going to actually digest themselves, which is what we refer to as autolysis. So if the enzymes that are carried by the lysosomes end up digesting that dying cell, we call that as autolysis. Then you sometimes also have some lysosomes in white blood cells that can actually be recruited during the process of inflammation. We'll talk about inflammation later on. And these inflammatory cells can actually spill out these chemicals and digest these infected cells. We call that as heterolysis. So remember that necrosis is going to be occurring in cells that have undergone that irreversible injury, and it's going to be as a result of either denaturation of the proteins that are present in the cell, as well as digestion with the enzymes of the injured cells. So how do these necrotic cells look like? So in the previous class, we talked about
the cells that are that have undergone reversible injury and some of the changes that we see in reversible injury. But here in necrosis, remember that there will be changes that are happening in the cytoplasm. There will be changes that are happening in the nucleus, and the different membranes are going to be affected. So you remember that there's going to be an increase in eosinophilia. Again, if you don't know what that is, check out the playlist, check out the introduction to pathology. And this is only because there's going to be a lot of eosin that's going to be binding to the denatured cytoplasmic proteins, and in part to loss of the basophilia, because the nucleus is being destroyed, the RNA is also being affected, the nucleic acids are being affected. The cells are going to take up this glacier-like appearance, and generally most of them are going to lose the glycogen particles that they store. Myelin figures will begin to become more prominent. The cell may become vacuolated and have these vacuoles that will appear like as if it's moth-eaten. And when you look at it under the electron microscope, you're actually going to see these discontinuities of the plasma membrane and even the organelle membranes. There's going to be marked dilatation of the mitochondria. They may sometimes even have these amorphous densities. You're going to have a disruption in the lysosomal membrane and even some intracytoplasmic myelin figures. And the dead cells usually love to attract calcium. We'll talk about intracellular accumulations. One of it is what's known as dystrophic calcification, where calcium deposits in dead tissue. Then you may also have some nuclear changes. DNA is going to be broken down. The chromatin is going to be broken down. Remember, clumping of DNA is not a feature of the irreversible injury. It usually happens earlier on in the stages of reversible injury. But then here in irreversible injury, the cells actually, the nucleus is going to begin to shrink. So that's known as pycnosis. It's going to become very, very dark. So there'll be an increase in basophilia because the DNA is going to be condensing. Then eventually this pycnotic or this shrunken nucleus is going to be fragmented, a process that we call karyorexis, which happens in about 1 to 2 days. And then the nucleus of many of these dead cells may actually completely disappear. And even when you look at it on the electron microscope, then you're able to see these nuclear changes, you're able to see this nuclear dissolution. Remember that the necrotic cells may sometimes remain in the body or where they are for some time, or they may be digested by enzymes and they completely disappear. So the dead cells are going to be replaced by these myelin figures, which are going to be either phagocytosed by other white blood cells or even further degraded into fatty acids. And these fatty acids, like I said, tend to attract calcium salts, and calcium ultimately is going to deposit in these dead tissues. There are different types of necrosis you need to be wary of. One common one is known as coagulative necrosis, also referred to as coliquative necrosis. This is characteristic in organs that are having their blood supply being cut off, so organs that are undergoing infarction. So you get these areas of necrosis in solid organs. The only organ where this type of necrosis doesn't happen is in the brain, because you have a different type of necrosis that's known as liquefactive necrosis, where the tissues begin to liquidate. So here, remember, you're cutting off the blood supply. Whenever blood supply has been cut off to an organ, the tissues begin to die, the enzymes are denatured. These enzymes are very important because they're going to be happening in the process of autolysis and breakdown of certain components in the cell. So if these enzymes are denatured, it means for several days, the architecture of these tissues is going to be preserved. And the affected tissue actually takes a very firm texture, and you may have these enzymes that are denatured. They can't really autolyze these cells, so there's just going to be eosinophilic, even anucleate cells that may be there for like weeks or days. You're just going to notice that some of these cells don't have a nucleus. A lot of them don't have a nucleus, but they have a normal architecture. Then you know that these cells have already died by coagulative necrosis. Very common in the myocardium. Then the white blood cells are going to be recruited to this site of necrosis, and then they'll end up digesting the dead cells by the action of their lysosomal enzymes, and the debris is going to be removed by a process known as phagocytosis. So here's an example of an area of necrosis here, this area that is wedge-shaped and it's yellow in color. This is seen in the kidneys. So this is a section of the kidneys. So this tissue had actually died. So if you actually look at this histology here on the right on image B, we can actually see that you can see here you have these glomeruli that are nicely formed and the normal appearance of the kidney on this side with a lot of these nuclei. While on this side, there is loss of this architecture. You can't really see some of the glomeruli, and some of these cells don't even have nuclei in them. And there are a lot of these cells in the interstitium, a lot of these inflammatory cells that you can see in the interstitium. So usually this is what's going to be seen in necrotic type of cells. The next type of necrosis is known as liquefactive necrosis, which is a special type of necrosis that often happens in the brain when there's infarction in the brain. So here it's also associated with some sort of infection. It could be a focal bacterial, even sometimes a fungal infection, because these microbes actually stimulate the accumulation of the inflammatory cells and even enzymes that are going to be digesting these leukocytes that are going to be digesting the tissues. They liquefy the tissues. So for some reason, if you get this hypoxia inside the brain, you get this blood supply that is cut off. For example, someone develops a stroke, that area is going to be liquidated, such that in the initial phases when someone develops a stroke, when you look at the CT scan, it may actually even be normal in an ischemic stroke. But later on, because now this area has been liquefied, the volume of there has gone and has been replaced with something else, you may see these changes later on. So remember that the dead cells are completely digested and they transform the tissue into a liquid viscous mass, and eventually this digested tissue is going to be removed by the phagocytes, and then this process will initiate this acute inflammatory response. And if there's a superadded bacterial infection, or there's a bacterial infection that is there, you may have this creamy yellowish substance which we refer to as pus. Here's a picture of liquefactive necrosis happening in the brain. Another type of necrosis is known as gangrenous necrosis. So this one here is not really a distinct pattern of death, but usually it's often happening in a limb that blood supply has been cut off. So usually common in the lower limbs. So it's a type of coagulative necrosis that usually involves many different types of layers of the skin. So when you have this bacterial infection that is superadded, then this coagulative necrosis is actually going to be modified to a type of liquefactive necrosis because of the bacterial infection that is there. When this happens, you refer to that as wet gangrene. If it's just blood supply that has been cut off, the tissues are dying, they're going to appear dry, there'll be the desiccation of the tissues, they'll be black in appearance, and you call that as dry gangrene. But if there's a superadded bacterial infection, you refer to that as wet gangrene. Then another type of necrosis is known as caseating necrosis or caseous necrosis. There are a lot of food references in terms of pathology. So remember that caseating necrosis is often seen with TB. So you get this area here, as you can see on the lungs, that looks like cheese. So this yellowish whitish cheese-like debris. So it's often associated with TB infection, and caseous just means cheese-like. And you have this friable yellowish whitish tissue that's going to be appearing. Under the microscope, you see a lot of necrosis happening in this area. You may see a collection of fragmented or even lysed cells. You may see this amorphous granular pink appearance. It's quite common on hematoxylin and eosin stains. Unlike with the coagulative necrosis, here the tissue architecture is going to be distorted. The cellular outlines, you won't be able to discern them. And of course, the area of necrosis is going to be enclosed by a distinct border of inflammation, as you can see here. And usually this is characteristic to keep the focus in this same area. And this focus of inflammation is what we refer to as a granuloma. There are different types of granuloma that we'll talk about in terms of inflammation. Another type of necrosis is fat necrosis, where you have these focal areas of fat destruction, typically because of release, for example, of activated pancreatic lipase into the substance of the pancreas or even to the peritoneal cavity. This is going to be happening in an abdominal emergency condition that's known as acute pancreatitis, where there's inflammation of the pancreas. Remember, it consists of these digestive enzymes that break down fats, digestive enzymes that break down proteins. So when you get these pancreatic enzymes actually leaking out of the acini and the ducts, they can actually liquefy the membranes of the fat cells in the peritoneum, and then the lipase can actually split the triglycerides esters containing within the fat cells. So when you release these fatty acids, they may sometimes combine with calcium in the process that's known as fat saponification, where you form this chalk-like white material. And this can actually even enable the surgeons, when they are opening up the abdomen, to identify the specific lesion. And the focus of necrosis is going to have this shadowy outline of the necrotic fat cells with basophilic calcium deposits that are going to be surrounded by an inflammatory reaction. The next type of necrosis is known as fibrinoid necrosis, common in blood vessels. So we see it mostly in immunological conditions, things like polyarteritis nodosa. So here you have this special type of necrosis. When you look at it under the microscope, because you have these immune complexes and these immune reactions that are happening where these complexes of the antibody and the antigen are depositing in the walls of the arteries, as you can see in this picture here. Then this deposited immune complex together with fibrin actually leaks out into these vessels, and it produces this bright pink amorphous appearance when you look at it under the hematoxylin and eosin, which are what we call fibrinoids. They are fibrin-like strands. So those are the different types of necrosis that you need to be wary of. Now let's get into apoptosis. So remember that this is different from necrosis because the necrosis is like genocide. A lot of cells are killed. Apoptosis, on the other hand, is like suicide, where individual cells are dying. So remember, the word apoptosis means falling off. So it's just a pathway where the cell is actually going to be activating certain enzymes that are going to be resulting in its own mechanisms that are leading to its DNA being degraded, the nucleus being degraded, the cytoplasmic proteins being degraded, and eventually the cell dying off. So it's also referred to as programmed cell death. Remember that the plasma membrane that we see in cells that are apoptotic is going to be intact. Ultimately, this is very important because if you don't expose certain components that are there on the inside of the cell, you won't stimulate an inflammatory response. So most of the times, apoptosis may not be associated with inflammatory response, as opposed to the cells that die by necrosis, which release their cellular components that could activate the inflammatory response, and such that necrosis invariably will always be associated with some sort of inflammation. So when we actually stain these cells with hematoxylin and eosin, we may see that the nucleus of these apoptotic cells is going to be in varying stages of the chromatin condensation and even aggregation, and ultimately karyorrhexis, that's the nuclear fragmentation. So your cells are going to be rapidly shrinking in size. They'll form these cytoplasmic buds. Eventually, they'll fragment into these apoptotic bodies that are going to be these membrane-bound vesicles of the cytosol and different organelles, because this cell has killed itself and packaged itself into these nice vesicles. And then these fragments are going to be extruded, and then there can be phagocytosed even without stimulation of the inflammatory response. So remember, apoptosis, like I said, is going to differ from necrosis. Apoptosis and necrosis can happen at the same time, but remember that apoptosis could either be physiological or pathological, whilst necrosis is almost always pathological, if not always pathological. Here are the large differences between apoptosis and necrosis. In terms of the cell size, it usually shrinks with apoptosis. In necrosis, there's some cell injury, there's increased osmotic damage, so usually the cells are going to begin to swell in size. In apoptosis, there's fragmentation of the nucleus into nucleosome-sized fragments, and in necrosis, there's pycnosis, which is shrinking of the nucleus, then karyorrhexis and karyolysis of the nucleus. The plasma membrane is going to remain intact, though there may be some orientation of the lipids. Some of the lipids are flipped, such that the lipids that are found on the inside of the plasma membrane will now be flipped to the outside so that they can sort of like stimulate those white blood cells for opsonization to make phagocytosis much more effective, and so that the immune system is not really activated in the process. Then, in terms of necrosis, the plasma membrane is disrupted. Cellular contents are going to be intact and they're going to be packaged in these apoptotic bodies. Whilst in necrosis, you have some enzymatic digestion of the cellular components that may actually even leak out of the cell. Apoptosis doesn't have inflammation involved, and there is some underlying inflammation that is seen in necrosis. You get this inflammatory zone. So it means apoptosis could either be physiological or sometimes pathological, whilst necrosis is always pathological. Apoptosis often tends to affect one or a few cells because it's like the cell killing itself. And in terms of necrosis, it's going to be affecting many of these cells and they are going to be killed off passively. So remember that apoptosis actually occurs in many normal situations and can actually be serving to actually even eliminate cells that are potentially harmful. If a cell is infected by a virus, it knows that something is wrong with me, kills itself off before the virus can spread, kills itself off before cancers can actually set in. So there are some points during the cell cycle where the cell actually checks for certain damages in the DNA, and if the cell has actually been damaged beyond repair, this will trigger certain proteins to be made, and these certain proteins are going to then stimulate the cell to kill itself through apoptosis. Like I said, apoptosis may be physiological or pathological. Physiological instances, it could be during embryogenesis, for example, development of the hand. So remember that when the hand is forming and the fingers are forming, they usually form as one structure. So let me see if I can just draw something for you here. So let's say this is going to be the bud of the hand. then these cells that are in between here are supposed to die off so that your fingers can actually form. So these cells are going to be dying by apoptosis so that your fingers and your thumb can actually form. The other thing is due to involution of these hormonal-dependent tissues, things like the endometrium during the menstrual cycle when you withdraw certain hormones and menstruation actually happens, even regression of the lactating breast after weaning off, or even just the prostate undergoing atrophy after a patient has been castrated. You may also have the cells in the intestinal crypts. For example, you want to maintain a constant number in these intestinal crypts. You want to maintain a constant number of cells in the skin so that it doesn't thicken. These cells need to be lost and they need to be shed off, so they need to die by apoptosis. It also eliminates cells that actually have served their purpose in the immune system. For example, neutrophils that have been activated in an acute inflammatory response. Leukocytes at the end of this immune response, because the only cells that need to remain are the memory cells, such that if that virus or bacteria has infected you again, you quickly divide and you can form other white blood cells. So you don't want to overload again the total number of white blood cells you have into your system. And also remember, in the process of stimulation of or training of your immune system to recognize self and non-self, this is the basis of autoimmune conditions. Because remember that if a white blood cell is not able to determine that this cell is part of us and it's not an enemy, it will end up killing even the normal cells, and this is what is going to result in these autoimmune conditions. So the cells that are able to react to self-antigen are actually killed off in the thymus during the process of training and maturation of the immune system. In terms of pathological apoptosis, if you have DNA damage that may be due to radiation, cytotoxic anti-cancer drugs, thermal injury, that's extremes of temperatures, and even hypoxia, you can have accumulation of misfolded proteins that may lead to endoplasmic reticulum stress. These are going to be causing pathological apoptosis, or even cell death in certain instances like viral infections like with hepatitis, and even death of cells that is mediated by T cytotoxic lymphocytes, or even sometimes spontaneously. It could be sometimes pathological atrophy in parenchymal organs after the duct has been blocked. For example, if the pancreatic duct is blocked, or the ducts that are draining the parotid gland, or even the kidneys, certain things can lead to atrophy and can lead to apoptosis of certain cells. Then you may actually see this in the cells when they undergo neoplastic transformation, they'll kill themselves. These are types of pathological apoptosis. So what exactly happens during the process of apoptosis? So there are some enzymes which are known as caspase enzymes. The reason why we call them caspase enzymes is because, number one, they are going to be cysteine protease enzymes. So they're going to be consisting of cysteine and they're protease enzymes. Number two, they're going to be cleaving individual proteins after the aspartate subunit. So that's why we call them as caspases. That's why we call them as caspase enzymes. So these are going to be activating a cascade, which in turn are going to be causing an imbalance between the factors that protect your cells from killing themselves and the factors that are going to be stimulating the cells to kill themselves. You're going to be having more of these pro-apoptotic proteins that are activated, and the anti-apoptotic proteins are going to be inhibited, and such that the cell can no longer stop itself from committing suicide, and it ends up killing itself. There are two important pathways that are necessary for this. There is the extrinsic pathway, which is known as the death receptor pathway, and there is the intrinsic pathway, which is known as the mitochondrial pathway. So before I actually come to this, I want to actually show you this particular picture, because I find it much easier for me to explain using a picture, then we'll come back to the words. So I'll begin with the extrinsic pathway because it's quite easy. So whenever cells are infected or they have noticed that there's a problem in them, they express certain receptors. So they could be expression of the tumor necrosis factor alpha receptor, they could be expression of the FAS receptor. Now, these receptors are going to be binding to a certain ligand. It could be binding to the FAS ligand, binds to the FAS receptor, or even the tumor necrosis factor alpha, or tumor necrosis factor rather, receptor could be stimulated. Once either of these receptors has been bound to, it means you are now triggering the cycle of the death receptor, this pathway that is going to end up killing this cell. So this is going to result in activation of adapter proteins, which are going to be initiating or activating a special type of caspase proteins, which are known as initiator caspases. Now, the initiator caspases normally are going to activate executioner caspases, for example, caspase 8. So they activate these executioner caspases, which are going to now set off a cascade of other enzymes that are going to be activated, which are enzymes like endonuclease, which can cause fragmentation of the nucleus, and even certain other enzymes that can result in the breakdown of the cytoskeleton, and eventually this results in the cytoplasm being put into these blebs, which eventually form apoptotic bodies, and then these are going to be having these ligands that are exposed. Remember those phospholipids that I told you are flipped from the inner surface of the membrane and put on the outside for the process of stimulation of these phagocytes for the process of phagocytosis to happen. So that pathway is known as the extrinsic pathway. On the other end of the spectrum, you have the intrinsic pathway, which is known as the mitochondrial pathway. It may be due to certain stimuli like growth factor has been withdrawn from the cell. It could be maybe the DNA has been damaged due to radiation, due to toxins, even free radicals, or proteins have been misfolded. Maybe this may be due to endoplasmic reticulum stress. Remember, this is going to be sensed by a group of genes that's known as the BCL2 family of genes, or rather proteins. And then this BCL proteins include BAX and BAK, which are pretty much going to stimulate the mitochondria and increase the permeability of the mitochondria. Certain channels are going to be seen on the mitochondria, and this is going to result in leakage of cytochrome C, as well as other pro-apoptotic proteins, which can initiate the initiator caspases and then eventually feed into this other extrinsic pathway and result in activation of the endonuclease enzymes and even the breakdown of the cytoskeleton. Under normal circumstances, the BAX and the BAK are pro-apoptotic, whilst there are some regulators that are going to be inhibiting the process of the mitochondrial pathway. You have the BCL2 and as well as the BCLXL type of proteins, which are like going to be regulating and preventing apoptosis from happening. So let's get back to this. So like I said, starting with the extrinsic pathway, we have two main types of receptors, the type 1 tumor necrosis factor receptor and the Fas ligand, which is the CD95 receptor rather. So when these are bound to, they have this receptor in the cytoplasm has what is known as the death domain. So it's going to be interacting with adapter proteins. So we get a T cell recognizing the Fas receptor or binding to the Fas receptor, or you could get the type 1 tumor necrosis factor being activated. So this is going to result in the adapter proteins that are going to be activated via that death domain of this receptor. So this death domain is going to be activating other caspases, like for example, caspase 8. And then this caspase 8, which is an initiator caspase, is then going to be activating the executioner caspases, which are going to be triggering other enzymes like the nuclease enzyme to degrade the DNA in the nuclear proteins, and then other enzymes that are going to be also fragmenting the nucleus, fragmenting the cytoskeleton, and eventually fragmenting the cells. Remember that the cellular proteins, notably a caspase antagonist, is known as FLIP, which normally is supposed to be blocking the activation of the caspase downstream of the death receptor. Some viruses can actually produce certain proteins that look like FLIP. So once now the FLIP has been produced by this enzyme, it will block the activation of these caspase enzymes. And once these caspase enzymes have been blocked, this cell will be incapable of killing itself via the extrinsic pathway, so it tends to live forever. So remember that in many cell types, caspase 8 may actually cleave and activate the pro-apoptotic members of the BCL2 family, which is known as the BID, and this can actually even feed into the mitochondrial pathway. And in terms of the mitochondrial pathway, like I told you, whatever is causing the injury, be it withdrawal of the growth factors, be it some hypoxia that is there, there is the choice between the cells actually surviving and the cell dying is actually determined by the permeability of the mitochondria. Once the mitochondria has become permeable, it's going to result in leakage of cytochrome C and other proteins which could stimulate this caspase cascade and eventually trigger the cell actually dying. So when the cell's actually deprived of the growth factor, you may, or other survival signals, then they're exposed to agents that are DNA damaging. There may be accumulation of these misfolded proteins, and a number of these sensors are going to be activated. One of the sensors is the BCL2 family, which is known as the BH3 proteins. And remember, these are only going to be containing the, only the third of the multiple actually contain the domains of the BCL2 family. So they're going to be activating other two types of proteins which are pro-apoptotic, like BAX and the BAK, which are going to dimerize, and then these are going to form a channel on the mitochondria. These proteins form this channel where cytochrome C can actually escape into the cytosol and stimulate. But under normal circumstances, there are other anti-apoptotic chemicals like BCL2 as well as BCLXL, which are going to be preventing this from happening. So these are anti-apoptotic. So normally there's a balance between the pro-apoptotic and the anti-apoptotic. And once the cytochrome C actually leaks out, can activate the caspase 9, and then this can actually trigger other caspase enzymes downstream, and this is going to be leading to apoptosis. So this is what's happening. Remember that the mitochondrial pathway seems to be the pathway that's going to be responsible for apoptosis in most situations. So remember that the apoptotic cells are going to be phagocytosed much more vigorously, like I told you, because of those phospholipids that are flipped. So under a normal circumstance, you have the phosphatidylserine, which is found in the inner leaflet of the plasma membrane. And in these apoptotic cells, these phospholipids are now going to be flipped and they're going to be found on the outside, such that the macrophages will recognize this and they'll phagocytose this even much more vigorously. And the dying cells actually also release soluble factors that recruit phagocytes, and there's this prompt clearing of the dead cells, and even they undergo secondary membrane damage and they release their cellular contents, which sometimes can induce inflammation. Then there's sometimes apoptosis that is mediated by the cytotoxic lymphocytes. Remember that the surface of infected host cells and even tumor cells, they can release certain receptors that can sometimes be stimulated by the cytotoxic T lymphocytes. Once these cytotoxic T lymphocytes have been stimulated, they're going to produce perforins, which are going to be entering into the membrane of this infected cell. And in addition to this, they'll pour in certain enzymes, which is known as the granzymes, which are going to be these protease enzymes that are entering into the target cell and actually degrading the target cell. Remember that these granzymes can actually cleave the proteins at the aspartate residue and can also activate different cellular caspase enzymes. So in this way, you can have the cytotoxic cells actually killing off specific types of cells that are infected, and this is going to be happening by directly inducing the effector phase of the apoptosis without even engaging in either the mitochondrial or even the death receptor pathways. Remember that the cytotoxic T lymphocytes also can express a Fas ligand on their surface, and they may kill target cells that express the Fas receptor on them. Then towards the end of this lecture, I want us to just look at autophagy, which is simply self-eating. The cell eats itself. So you have lysosomal digestion of the cell's own components. So this usually happens when you have prolonged periods of nutrition deprivation. We are not getting enough nutrients. The cell is going to begin to eat itself. So these intracellular organelles and even portions of the cytosol are going to be sequestered in these vacuoles, which we call autophagic vacuoles. And then after that, it is actually postulated to be as a result or formed from these ribosome-free regions of the endoplasmic reticulum. So these vacuoles can then fuse with lysosomes to form what are known as autophagolysosomes, and some of these lysosomal enzymes will now begin to digest the cellular components. Remember that these digested components can be recycled in the cell for it to remain viable. So with time, if you continue starving the cell, they're no longer going to be coping with them eating at the cellular organelles, and then at this particular stage, this is what's going to trigger the cell to die. Remember that autophagy appears to trigger cell death that is actually quite distinct from the necrosis and the apoptosis that we actually discussed earlier on. And it's actually what we think may be happening in many of the degenerative conditions that are affecting the central nervous system. I really hope you enjoyed this lecture on cell death, on apoptosis and necrosis. If you did, consider subscribing to the channel, hit the bell notification icon so you never miss on such amazing content every time I post. To Zambia and beyond, my name is Dr. Moses Kazevu. Until next time, bye-bye.